Assalamu alaikum. Hope you guys are enjoying your vacations. My name is Munazza Shfiq and my topic is breeding process F1 and F2 formation in plant. Let's discuss the introduction of plant breeding. Plant breeding is a science of changing the trait of plants in order to produce desired characteristics. It has been used to improve the quantity of nutrients in product for plants and animals. The goal of plant breedings are to produce crop varieties that boost unique and superior trait for a variety of agricultural application. Plant breeding can be accomplished through many different techniques, ranging from simple selecting plants with desired characteristics for propagation to method to make use of knowledge of genetics and chromosomes to more complex molecular techniques. Gene in plant are what determine what type of qualitative and quantitative traits it will have. Let's discuss the history of plant breeding. Plant breeding is an ancient activity dating to a very beginning of agriculture. Probably soon after the earliest house breakage of cereal grain, humans began to recognize degree of excellence among the plant in their fields and save seed for the best for planting new crops. Such tentative selective method were the ancestor of early plant breeding process. The result of early plant breeding procedure were notable. Most present day varieties are so modified for their wild forefeathers of they are unable to survive in nature. Indeed, in some cases, the cultive forms are so remarkably different from existing wild varieties that is different even to identify their ancestor. The remarkable transformation were accomplished by the early plant breeders in a very short time for an evolutionary point of view, and the rate of change was probably greater than for any other evolutionary event. In the mid-18th, George Mantel outlined the principle of heredity using pea plant and thus provided the necessary framework for scientific plant breeding. As the law of genetic inheritance was further delineated in the earlier 20th century, a beginning was made toward applying them to the improved plant. One of the major facts that emerged during the short history of scientific breeding is that an enormous wealth of genetic variability exists in plant of a world and that only a start has been made in trapping its potential. Mendel law of segregation cross of pure plant and pure white flower stain of pea. Capital R stands for the gene for purple flower and small r for gene of white flower. Mendel's law of segregation cross of purple flower and white flower stains of pea. Let's discuss the method of plant breeding. There are two types of plant breeding method, classical and controversial. Classical plant breeding introduce new genes into a plant can involve using the same kind of plant, a different plant or different organism such as an microorganism. In classical breeding, thousands of genes are being rearranged where genetic engineering involves the specific handling of a single gene. Let's discuss the controversial plant breeding. Controversial plant breeding is the development or improvement of cultivators using controversive tool for manipulating plants genomic within the nature genetic boundaries of the species. Method for breeding cross-pollinated species include mass selection, family selection, and synthetic. Now, we discuss the generations. F1 generation and F2 generation. F2 generation and F1 generation. F1 generation means F first generation or the initial cross between two genetically distinct plants. Often an F1 cross do not yield the desired scores because some traits do not show up in those first generation. For example, the bee pod on one plant and all its offspring would produce green pod while another plant would produce all yellow pods. Once fertilized, the parental generation show green plant which contain the genetic information for the first generation of offspring or the F1 generation. F2 generation. The offspring from F1 generation comprises the second filial generation. The F2 generation is the result of a cross between two individuals. For example, when a round seed lines was crossed to a wrinkle, the F1 generation was all round and F2 generation phenotypic ratio 3:1. 3 is round and 1 is wrinkled. In plant or animals that cannot fertilize, F2 generation is produced by crossing F1s of each other. Here we look the parental round seed line crossed with wrinkled seed line. The F1 produce a round seed progeny and F2 produce phenotypic 3:1. 3 is round and 1 is wrinkled. From this we can conclude that round P in F2 generation consists of both types. To test this hypothesis, Mendel allowed round P plant from F2 generation to self-fertilize and secured the progeny from each plant. 1 out of 3 round P plant from the F2 generation were two breeding and produce only offspring with round Ps. The remaining 2 out of 3 produce a ratio of 3 round and 1 wrinkle. Now we will discuss the difference between F1 generation and F2 generation. F1 generation is the first filial generation of the offspring from the parents, but F2 generation is the second filial generation of the offspring generating through interbreed of F1 individual. The first generation is distinctly different from parental type, and F2 generation may exist some parental phenotype. Meanwhile, F1 generation is important in hybridization, obtaining the best characteristics of the parent in offspring. F2 generation is important in interpreting in order to maintain stable trait over generation. Thank you so much for